I will share my screen and hopefully, let's see. All right. Can you now see your slides? Not
Just
yet?
see the top of your screen.
Oh, all right. Let me then try to, let me share that one. How's that? Any better?
Yes, that's
There
me.
you go. That is you, Lisa. Well, welcome, Lisa. Thank you for joining us today. For the students who are still joining, if you would add into the chat your name and your high school and your favorite ice cream flavor, we will use that to take attendance. But thank you for coming on to talk with the students today about your career path. It is wonderful and fascinating, and I love all the images that you have here, and I'm eager to hear about them. But so Lisa, maybe if you don't mind, could you say a little bit maybe about what you are doing these days, and we'll
Mhm.
back up to how you got there?
Sure. So my name is Lisa Stanek. I am the Vice President of Translational Science at Finia Therapeutics, which is a biotechnology company. It's a private, so a small private biotechnology based out of Waltham, Massachusetts. And I think through this little session and some of the slides I prepared, I'll explain what that means. A lot of people don't know necessarily what translational science means, but I'll sort of walk you through my career journey, how I got to where I am, sort of my educational path, and then answer any questions that you might have. Okay. So just a little bit about myself personally, so you get to know me a little bit better. I grew up on Long Island, New York, so I love the beach. I'm definitely a beach person. Um, Here's some pictures of my family. That's my husband, Grant, and our 4 kids. So I have a 16-year-old, a 15-year-old, a 12-year-old, and an 8-year-old. So we're very busy, full household. And to make life more interesting, we just got a puppy. So that's our 8-month-old puppy named Charlie. He's a little Shih Tzu poodle mix that we added into the mix fairly recently. My hobbies are running. I love to run. Every fall, I usually do a half marathon or some 10Ks thrown in. And I'm actually a yoga teacher. So when I'm not doing translational neuroscience, I actually teach yoga. I actually teach yoga at work. So we have a gym in our really beautiful lab space in Waltham. There's a gorgeous gym downstairs, and it has a yoga studio. And so I teach my colleagues yoga so that we can all relax and calm down after our busy and stressful days. Um, so the beginning of my journey started in college. After high school, I went to Union College, which is a small liberal arts college in upstate New York. It's in Schenectady. I really liked biology. So in high school, I took AP biology. We dissected pig hearts, and I think we dissected a rodent. I was just fascinated. I thought it was so cool. I just loved like all of the experiments, like the hands-on experiments that we did in our AP bio class. So I majored in biology in college. And my junior year in so yeah, my junior year in college, I did an internship with my neuroscience professor. I really loved his class, and he's like, "You're super interested. You love the lab component of the class. Would you want to do a summer internship with me?" And I said, "Of course, that'd be super fun." I got a paid stipend. I got to stay up in upstate New York for the summer, and it so happened that he was interested in studying dragonfly electrophysiology. So really understanding how dragonflies capture their prey. Dragonflies are expert hunters, and they almost look like airplanes, like fighter jets. And so I spent 2 years in his lab studying dragonfly electrophysiology, so action potentials in the dragonfly brain. They do have a very primitive brain, and the setup looks like that the one below. Lots of wires. It's really like almost like an electrical engineering setup. but I really loved it. And at that point, I decided I was going to go to graduate school. So before then, I thought about medical school. I thought about either becoming a medical doctor or even a veterinarian. I love animals. But with the research that I was doing in the lab, it became clear I really liked research. I loved asking questions and doing experiments. I loved being in the lab. And so he prompted me. I was so lucky to have such a wonderful mentor. He prompted me to go to a PhD program. It hadn't even crossed my mind. I didn't even think about it. But so I started looking into graduate school specifically for neuroscience programs. And I ended up going to Emory University, which is in Atlanta, Georgia. It's an amazing, beautiful college and university. If anybody is interested in going to school down south, highly, highly recommend it. The best 6 years of my life spent in graduate school, getting my PhD in neuroscience. While I was there, I moved away from invertebrate electrophysiology and I moved into mammals. And I studied in graduate school. I actually studied non-human primates. I did research in the Yerkes Primate Research Center, studying social behavior in monkeys, and also did quite a bit of research in mouse genetics. And the company that I work at now, and I'll tell you a little bit more about that in the follow-up slide, we do gene therapy. So some of you may have heard about gene therapy. We engineer viruses to deliver genes, replace genes that are missing in patients that have diseases. So that's the work that I'm doing now. And I'll in the next slide, I'll sort of explain a little bit more about that. And stop me if you have any questions. So I already told you I started out my career at Union College. I got a bachelor's in biology, and that led me to my PhD at Emory University. Every PhD program is different. Every PhD student's career trajectory is different. Some people take 4 years to finish their PhD. Some people take 8. My roommate in graduate school took 8 years. So it just depends on your project, your advisor, and your luck. And so I was actually really lucky. I had a great advisor. I had a great project, and I finished in 5 years, which in the grand scheme of things is actually pretty short. But like I said, it was the time of my life. I loved every minute of it. I made some of the best friends that are still my best friends today in graduate school. So you may look at these numbers and be like, wow, that's a long time to be in school, but not when you're really loving it. So graduate school was great. And typically after you get a PhD, it is very traditional to do a postdoctoral fellowship. So that's like kind of further training. It's sort of akin to a residency that you would do after graduating medical school. So I did my postdoctoral fellowship at Harvard. That's what brought me here to the Boston area. And I did that for 2 years. And I knew, even in graduate school, I knew that I wanted to work in pharmaceutical drug development. I wanted the things that I was doing in the lab to really translate to human disease. I wanted to help people with the research that I was doing. So after my postdoctoral fellowship at Harvard, I got my first ever job. At this point, I'm in my 30s, which seems crazy that at 30 is when I got my very first job, but I wouldn't change any of it. All of the training, the education, Emory and Harvard. Not only was it really important for my career development, but it was fun. And like I said, I had a good time of my life. So I got my first job at a company called Genzyme. This company no longer exists, unfortunately, anymore, but it was an amazing company that was built upon the premise of treating patients that had rare diseases. So patients that have rare disease are often overlooked by drug companies because they feel that there's not enough market for their drug, not enough people to take it, not enough money to be made. But Genzyme really changed the business model. And we were treating predominantly rare diseases, and actually a very profitable company, making a profit. About 3 years into my time at Genzyme, we were acquired. This is a common thing that happens in biotechnology in the field of pharmaceuticals. companies kind of eat up, it's like Pac-Man, they kind of eat up little companies. And so we were acquired by a company that you've probably all heard of, one of the top 5 pharmaceutical companies in the world called Sanofi. And I continued my career. So the work that I was doing in rare disease and specifically in gene therapy, trying to treat rare diseases with gene therapy, I continued on and I stayed there for 14 years, which is a very long time in the world of biopharma. People kind of hop around every couple of years, but I really loved what I was doing. I loved my team. They were like my family at that point. So I stayed there for 14 years. And about 4 years ago, it was right in the middle of COVID, a company called Affinia, a brand new sort of budding gene therapy company, reached out to me to lead their preclinical and translational science program. And it was a really fantastic offer. And I would be getting to work, with new people. And so I've been now at Affinia Therapeutics for the last 4 years as the VP of Translational Science.
It's amazing. Can I just ask you one thing about your
Sure.
career path? I love the way it's drawn out here. You said Genzyme was your first job, but the schooling that you did, you were not paying for going to school, right?
Yeah, that's a really great distinction. I think it's important. I would be so broke if I had to pay for all of that. I did not know. So after college, for a doctoral program in the sciences, they typically pay your tuition. So Emory paid my tuition, and they give you a stipend. So it's essentially a salary, a monthly salary, just like I get now for a job. It's not, you're not rolling in dough. It's not a ton of money, but it was, I was a 21-year-old girl. Like I didn't need a ton of money. And it was plenty to pay for my apartment and my food and overhead. And same for postdoctoral fellowship. So at that point you get started to make more money. It's still a stipend. And then it was Genzyme where I received my first real paycheck. So yeah, you're not accumulating student debt at that point.
Yeah. It was something that surprised me as well when I was going and getting my PhD, right? That suddenly you're going to be getting a stipend and your tuition is paid for by the university. So,
Yeah,
or by the
yeah. And for someone who loves school, I mean, it's too good to be true, right? Like they were paying me to go to school.
right.
And so why would you not do that? Like it was, for me, it was a no-brainer.
Yeah,
Um,
great. Thanks for distinguishing that. Let me move on. Yeah, translational science.
yeah. So what's translational science? I often tell people what I do and they like nod and smile, but I don't think a lot of people really understand. So if when it's laid out like this, kind of visually, I think it makes a lot of sense. So it sort of sits in the middle between basic science and research. Sometimes we call it discovery research, and the clinic. So I will take the discoveries that come out of the basic research labs and translate them into a drug and then hand them off to my clinical colleagues. So within a company, like my current company or at Sanofi or Genzyme, there are a lot of people doing basic science, trying to innovate, develop novel drugs, in my case, to develop novel viral vectors. And I implement them in terms of a drug. So what are the diseases that we could treat with this new entity? What are the animal models that we need to show efficacy or safety in? And then once I've shown that this new drug works to treat a disease in an animal model and is safe, then it gets handed off to my clinical colleagues, the medical doctors, that will put it into a human clinical trial. So when I brought my kids to work, we had a bring your kids to work day, and I explained it. They're like, oh, you do clinical trial in animals, which is pretty accurate. So everybody knows what a clinical trial is in humans and patients. I do it just in the mice and the monkeys. So if you go to the next slide, this is my job and my job description in a snapshot. So the things that I have to do, I have to do model development. So that means, are there animal models of this disease that I'm studying? If so, what's wrong with them? Well, how they are not patients. They can't tell me like, oh, I'm having trouble walking, or my back hurts, or I'm having like, brain fog. So I have to use a number of behavioral testing tools in order to understand what's wrong with our animals. Because once I can show that these animals are sick, they have some kind of what we call a phenotype, then I can treat them and show that our drug improves this deficit. Another thing that's a big part of my job is how do we deliver the drug? So not all drugs can cross the blood-brain barrier, for example, or not all drugs can go to the organs that we need them to go to. So part of my job is to understand delivery. How do we give this drug? For some of the neuroscience diseases that I study, the neurologic diseases, sometimes you have to deliver it directly to the brain. So that involves brain surgery. I've probably in my lifetime done hundreds of thousands of brain surgeries on small animals. And, believe it or not, patients that have severe neurodegenerative diseases are not averse to having brain surgery in order to deliver these drugs. So sometimes we do that. Ideally, we just give an injection. So something like when you get your COVID shot or your flu shot, we would give a systemic injection, but it all depends on the drug and the properties of the drug. A very big and very important part of my job is to understand the therapeutic window, and that's really about safety. So understanding what's the highest dose of the drug that we can give that is safe, because first and foremost, we don't want to harm the patients. We want to help them. So understanding safety window, and then, tolerability and scalability. So, mice are not people. Most of the work, the preclinical work, is done in mice, but human organs, human brains are 1,000 times the size of a mouse brain. So we do have to scale up and make sure that these drugs will still work in larger species. So my work starts in mice, but it usually moves up to non-human primates, because we want to make sure that this is ultimately going to work in a human, and that's the closest species that we have. So in a nutshell, this is my job description. This is what I'm expected to do at my current company. If you go to the next slide, this is just some more. That's my team. That's the translational science team. And just some of our roles and responsibilities in the company. So in addition to the previous slide, which is showing, we lead the non-clinical, so the preclinical or non-clinical development strategy. We drive the execution of all that research, the animal research, through what's called an IND. I don't know if you guys have heard that term before. An IND is an investigational new drug, and you have to file with the regulatory agencies, the federal government, an IND that they have to review carefully and approve if you put anything in a person. So that's why, you may like hear things, or, oh, that's not FDA regulated, like these homeopathic medicines that you could buy at CVS. It's not FDA. That means that nobody has provided like a significant data package to the FDA and hasn't gotten the gold stamp of approval. To do any kind of human clinical trial, you have to get FDA clearance. And so you could do that by submitting an IND. Another part of my job is indication selection. So that's defining the diseases that we want to study. And this all depends on what kind of molecules, what kind of viruses are being developed by that basic research team. So they sort of hand me something and they're like, what can you do with it? And depending on the molecule or what we're talking about, it's like, I could treat Huntington's disease with this, or, this is maybe a treatment for Parkinson's disease. So depending on what, the basic research team discovers, I will implement it into a disease indication. I work not alone. We work in a huge network. I leverage a lot of academic partners. So we have to appreciate that we're not the only ones studying this disease, nor can I be an expert at every disease under the sun. So a big part of my job is identifying key opinion leaders or people in the field that really understand this disease really well, and forming relationships. I also support our business group at the company. So we are a company. We are not a non-for-profit. So we do, need to make money to earn revenue. And part of that is by forming collaborations and partnerships with other companies. So I might have a great virus that treats the kidney, but we don't really have the bandwidth to have a kidney program. So I might sell that virus to another company. So that's part of how drug companies earn revenue and make money. I write papers, so I publish papers, I publish patents. So a big part of my job is a lot of writing. So probably the part of my job I like the least, to be honest. I find it really boring and time consuming. I love doing the research. I love crunching data. This picture of me in the lab it's almost a joke because I probably step foot in the lab maybe once a year, if not even more infrequent. But I wish. I just don't, unfortunately, have the time, and I'm lucky to have a team of wonderful scientists that can execute the work for me. A better part of my career, I looked like I did in this picture. So I spent most of my time as a scientist in the lab doing animal surgeries, making buffers, like running assays. It's only probably in the last like 5 or 6 years of my career I've had more of a kind of a leadership administrative role. And then I present my data at meetings. I'm going to a conference. Another thing that people might not mention about grad school and postdoc, like having a career in science, is you get to travel a lot. So in my time at Emory, I probably visited 4 different countries, gave talks all over the world, traveled all over the United States with my friends. Again, like I said, really best years of my life. And definitely an upside of being in this field is, as a grad student, we were poor. We didn't have a lot of money, but I was traveling all over the world because the university paid for my trip because I was presenting research. I think there's maybe one more slide. And so this is just a snapshot, a day in the life at Affinia. We're a super fun company. We do lots of team building. It's a small company. We're about maybe 70 people right now, but we have a lot of fun. We have a very tight-knit team. There's a beauty in being part of a small company because there's so few of us. Everybody is really close, and we work really hard, but we also make sure to have a lot of fun. So
That's amazing. I love your story and I love the trajectory of it. I have some big picture questions and then a couple
sure.
of particular questions. I mean, one thing that I hear you saying as a theme is that it really is about the relationships that you've built along the way and that this is a team effort. And that, grad school was fun because you were with people that you were compatible with, that all love science, that we're all, you were doing it, if not on the same project, but at least in a community, right?
Yeah.
And that what you
Yeah.
like about your current company also is that it's a community, which I think is maybe not what people immediately think about when they think about a career in science as a very team-oriented effort, or at least a lot of network and connections that are super important as you go through. But that certainly seemed to be some of what you were saying through your career.
Yeah, I think people think of a scientist like a nerdy scientist, solitary in the lab in a dark, lab. That's not the case at all. You can't be successful in a vacuum. You have to collaborate a lot because you can't be an expert at everything. The person working in the lab next door to you may actually have, know how to use this really cool microscope, or maybe they have experience to doing this behavioral test. So you have to be part of a community, and that's how you become successful.
Yeah, I love that. We certainly emphasize collaboration within this program, but I think it can't be said often enough that, yeah, you can't know everything. Who your neighbors are and who your network is will continue to improve the science
Yeah.
going forward. I had a question about your the overall approach. It sounds to me, maybe this is right or wrong, you can correct me, that you don't go after particular disease targets, but rather you do discovery of molecules, viruses, entities that could then be applied to any number of disease targets. Is that accurate? I think of a lot of other
Yeah.
companies saying, we're going to cure diabetes. We're going to address, I don't know, some rare disease, a kidney disease, right? But it doesn't sound like
Yeah.
that's what you guys are doing.
No, it's not at all. And that's a really interesting distinction that you identified. So we're a virus company. Sometimes you might hear of it as like a platform company, like building and developing novel viruses are a platform. So as the translational scientist, and I've told you I'm a neuroscientist by training, I have to go where the viruses take me. And currently, right now, they've taken me to the heart. And even though I'm a neuroscientist, the brain is my comfort zone. I am parading around as a cardiologist these days because our viruses are really good at infecting the heart. And so I've been studying over the past year cardiomyopathies, diseases that affect the heart, rare genetic diseases that cause people to go into heart failure. So currently with my company, absolutely, I go where the viruses take me. Other companies, you're right, might be an oncology company and they are going after a certain cancer indication, and they look for drugs that treat that indication. It's just a different philosophy. At
Yeah.
Sanofi, we were very much focused on certain disease indications. And so we threw everything but the kitchen sink at them. Small molecules, viruses, antibodies, whatever we had in our arsenal, because we were really focused on the indication. But at my current company, we're more focused on the virus and what the virus can do for different patient populations.
Yeah.
So
No.
yeah, it's a different philosophy.
Yeah, it's focusing on the modality, which I think is really, really interesting,
Exactly.
very, very
Exactly.
productive. Um,
Yeah.
there's one last thing that I just want to point out to the students, and I do want to give them a chance to either put a question in the chat or ask you directly. In that picture that you showed, you guys are gowned from top to bottom.
Mhm.
We spend a lot of time in the lab, but we do not
Mhm.
gown in quite that same way.
Yeah.
Can you just maybe unpack
Yeah.
a bit about the gowning that you do and why?
Sure. Yeah, that picture was taken in our animal facility in the middle of a necropsy. So in the laboratory, our analytics lab, for example, we would just be wearing a lab coat and goggles, and that's it. When we are taking out brains and hearts and guts and livers, oh, we like to dress from head to toe. So yeah, it was an all hands on deck necropsy. I think we were taking down 98 animals over the course of a couple days. So, and yeah, it's not glamorous. We were all sweating under there. It's hot in there, but there's a reason we wear all that protective. We just don't want to get our street clothes dirty.
Yep, definitely. That's
Yeah.
awesome. Thank you. I mean, it's a fascinating job that you do. It's a fascinating career path. It sounds like you were always interested in life science and that you have found different ways of applying your interests and your talents. We've had a lot of people come through and say that they originally wanted to be vets, but you are the first one that continues to work with animals in some fashion.
In a slightly different capacity.
It is, yes. But
Yeah.
we've had, yeah, people who run vivariums, right? There are veterinarians who run
Mm.
vivariums for companies. And yeah, so and the last week we had a gal whose dad was a taxidermist, and that's what got her involved and
Oh,
is
Sam.
yeah,
I know. Yeah.
interested in science. So yeah, all the folks that love animals and love helping, either animals or people, there is a lot of room in science to do all that.
Absolutely.
Um, do before we let you go and we turn to our math lesson, and I love that you said that you spend some of your day making buffer. Last week on this, we made buffers, and we're going to be pHing buffers
Oh,
on Saturday.
a great
So
skill
yeah,
set.
we do a lot of that, right? Are there any questions that any of the students have? Oh yeah, Ruby's asking what the favorite part of your job is.
My favorite is getting data and analyzing it and having it look the way I expected it to. I can't, I've been doing this for like 20 years. And just last Friday, we got some great data. We essentially treated an animal model of disease and they got better. And for 20 years, I've been doing this and it never fails to make me so happy and so excited. And so really, seeing a drug effect, a positive one, the one that we expected, is still so exciting.
That is it's such a great moment when you do get the data and you can look at it
Mhm.
and it all clicks together. It's like all the puzzle pieces falling into place at once. And I think some of it is that it doesn't happen all that often. It's not always what you want it to be. And so when
It's
it does, you're like,
true.
it's awesome.
It's
Um,
absolutely true.
yeah. Parnov is asking, what drugs have you produced so far that have worked? Are there any that are that
That's
you
a
can talk
really
about?
good question. If you ask 100 scientists that question, probably 99 of them would say none, because drug development is really hard, and um, probably only less than 1% of drugs make it to the market, like where they could be utilized by patients. I'm really fortunate. I worked on a drug, a gene therapy drug, back when I was at Genzyme for spinal muscular atrophy. So if you guys want to Google that, it's called Zolgensma with a Z. And patients with SMA or spinal muscular atrophy don't survive to their second birthday. The gene therapy drug that we developed for them, there are patients, it's standard of care now, and there are patients that are, I think, 8 years old
Mhm.
and still alive and happy and healthy and running around with their parents that would not have survived past their second birthday. So it's what keeps me going.
Yeah, that's an amazing story. Congratulations. What a wonderful impact. And yes, it is so very rare. Well, congratulations. It seems like a wonderful career. I just came from a meeting today where there was a lot of talk about AI. I would be remiss in not asking if you guys apply AI or are thinking about AI as it applies to biology, because I think these students are going to see AI and biology talk to each other in ways that we can't really know just yet.
Yeah, you guys are all really lucky, and that I didn't have AI at my disposal, most of my career, but we absolutely use AI in my company. A part of what we do is design novel viruses. And by changing small, like single nucleotide sequences on the virus. And so we use a lot of generative AI in order to help us with bioinformatics and development of these novel viruses.
Awesome.
Computers are really, really smart and really good at stuff like that. So we definitely utilize a lot of AI.
Uh, it's so true. It's not just for writing bad poems or drawing crazy.
Chat
Right.
GPT.
So,
Yeah.
Lisa, it's been wonderful to talk to you. Thank you so much for taking time out of your day. You're very busy. You have a lot of balls and a lot of plates spinning, right? So a lot of balls in the air and a lot of plates spinning. So I really appreciate you taking time to talk to the students of this.
Great. Thanks. And if anyone wants to reach out, you can provide them my contact information. I'm happy to chat with them.
Awesome. Thank you so much.
Okay.
Thank you.
Thanks. Take care. Bye.